morning. My name is Christy Abraham, and I'm a rising senior at Nova Southeastern University, majoring in biology and behavioral neuroscience. For the past 10 weeks, I've been working in the Lowry Lab, investigating the function of Wolff-Hirschhorn syndrome-associated genes in developing Xenophis laevis embryos. Wolff-Hirschhorn syndrome, also known as WHS, is a neurodevelopmental disorder that is primarily characterized by craniofacial malformations, heart and skeletal defects, intellectual disability, and seizures. The most obvious clinical marker of Wolff-Hirschhorn syndrome is the appearance of the Greek warrior helmet, which is characterized by a wide and flat nose, low-slit eyes, a high nasal bridge, and widely spaced eyes. WHS patients may also express micronephia, which is an undersized jaw, microcephaly, which is an abnormally small head, and defects to the eye and optic nerve. Currently, WHS targets 1 in 50,000 individuals, but a majority of these patients do not survive to reproductive age. Twice as many females than males are affected by Wolff-Hirschhorn syndrome, but this gender skew is not well understood. Diagnosis of WHS is often difficult, therefore it may be more common than reported. The average life expectancy is unknown because many cases of WHS vary in their severity. Now that we are familiar with what Wolff-Hirschhorn syndrome is, how exactly does it occur? It arises as a result of a mutation on the P arm of chromosome 4 in the 4p16.3 region. These mutations can be deletions or microduplications. Many genetic studies have sought to examine which exact genes are involved, and they've narrowed it down to these 4: WHSC1, WHSC2, LATM1, and TAC3. But the Wolff-Hirschhorn syndrome field still does not understand how either deletions or duplications in these 4 genes ultimately leads to the Wolff-Hirschhorn syndrome phenotype. Therefore, the goal of the study is to ultimately investigate the functions of these genes to see how the Wolff-Hirschhorn syndrome phenotype may be arising. We will do so by analyzing craniofacial morphology following combinatorial knockdown of these 4 genes. This will both inform our understanding of what these genes are and WHS as a disorder. So what do we know about these 4 genes? WHSC1 stands for Wolff-Hirschhorn syndrome candidate 1, and it was the first gene to be implicated in this disorder. It is also found to be deleted in a majority of WHS cases. We know that it is involved in many developmental processes, as well as regulating gene expression, but how exactly that functions in the context of Wolff-Hirschhorn syndrome still needs to be more understood. WHSC2, also known as NALPA, is involved in protein translation, mRNA processing, as well as cell proliferation. But again, we don't understand its developmental function in the context of Wolff-Hirschhorn syndrome. LATM1 stands for leucine zipper EF hand-containing transmembrane protein, and it's actually a mitochondrial protein. Now, we are all familiar with the fact that the mitochondria is the powerhouse of the cell and thus creates all of its energy. So this tells us that LATM1 is ubiquitously expressed in all cells. And it's very interesting to think how a mitochondrial protein may lead to this very specific neurodevelopmental disorder. And finally, we have TAC3. TAC3 stands for transforming acidic coil-to-coil protein 3, and this is a protein that the Lowry Lab has studied in depth. And while these other genes are involved in gene regulation and housekeeping, TAC3 is a regulator of microtubules. And microtubules are a component of cells which ensure cell movement. And it's interesting to think that if cell movement is occurring improperly during Wolff-Hirschhorn syndrome, then maybe TAC3 is implicated in the disorder. TAC3 is actually how the Lowry Lab first became interested in WHS because the lab has been studying this gene for years, and in doing so, we've learned that it's not well understood in the context of Wolff-Hirschhorn syndrome. Okay, so I've given you a brief overview of what these genes do, but how exactly do they result in the WHS phenotype? Well, during development, there are a certain group of cells called neurocrest cells, and these exist as part of the neural tube. They start as epithelial folds and eventually delaminate and migrate to different regions of the body. they become the facial structures, they become heart tissue, and they become skeletal tissue. And these are some of the regions of the body that are also affected by Wolff-Hirschman syndrome. And so this causes us to wonder if, in fact, errors in neurocrestall migration may ultimately cause the WHS phenotype. Now, we've established that neurocrestals are important and that the WHS-associated genes may play a role in neurocrestals. So how are we going to investigate this? The Lowry lab uses the frog model, Xenopus laevis, because its eggs are easy to collect and easy to maintain. In addition, its genome may be easily manipulated, and its craniofacial morphology is easily quantifiable. The first thing the Lowry lab did before I joined this project was to see if WHS-associated genes were in fact involved in neurocrestals. They did so by performing an in situ hybridization with the 4 WHS-associated genes along with Twist. And Twist is a well-known marker for neurocrestals. And so in an in situ hybridization, the mRNA that is normally expressed for each of these genes is labeled in purple. So what we're looking at here are stage 35 embryos of Xenopus laevis that are fixed, meaning that they are not alive. And you can see each side of the embryo with the eyes located right here. And wherever you see purple is where the mRNA of that respective gene is being expressed. Now, these arrows are pointing to a particular location called the pharyngeal arches, and these are where the craniofacial neuropress cells are migrating down to ultimately form the facial structures during development. Now, we've seen that by looking at these images, we can tell that at least majority of these genes are at least somewhat expressed in the pharyngeal arches, and this is very reassuring because it tells us that the WHS-associated genes are in fact involved in neuropress cell migration. To further examine whether WHS-associated genes were in fact involved in migrating neuropress cells, we knocked down each of the genes in separate embryos on one side of the body only. And this allows the embryo to serve as an internal control. We then performed an in situ hybridization in which we looked for a twist, or marked for twist, which again is a marker for neuropress cells. So in these lateral images, we see one side of the embryo, which is a control, and the other side shows the gene knockdown. And wherever it's labeled and it's stained in purple is where the neuropress cells are being located. Specifically, these are the pharyngeal arches, as I mentioned before. And in the controls, we can tell that the pharyngeal arches are very prominent, they're very dark, they're primarily located in the lower regions and migrate upwards. In the WHSC1 knockdown, though, however, we do still see pharyngeal arches, but they are shorter and primarily located in the upper regions. In the TAC3 knockdown, again, the pharyngeal arches are shorter, but they do exist in the lower regions like the control. However, in the LET-M1 and WHSC2 knockdown, we didn't see any significant changes in pharyngeal arch localization. So this tells us that knockdown of the WHS-associated genes does play a role in hindering the migration of the neuropress cells. So now that we've established that the WHS-associated genes were expressed in cranial neuropress cells, in neuropress cells as a whole, and that erroneous motility may cause the WHS phenotype, we wanted to look at a different feature, and that was in fact craniofacial defects. Several patients who have WHS primarily show these defects, which is aesthetically very common and noticeable. And so for the scope of my project this summer, we wanted to see if when we manipulate these genes in Xenopus, did they in fact show these defects as well? So how did we investigate this? First, we injected Xenopus laevis with HCG, which is a female reproductive hormone, to induce ovulation. Next, we collect, fertilize, and dejelly the eggs in order to prepare them for gene knockdown. We injected each embryo 4 times during the 2 to 4 cell stage with morpholinos. Morpholinos are antisense oligomers that suppress gene expression and ultimately prevent a protein from being translated. When they enter the cell, they bind to the complementary mRNA of interest and ultimately suppress gene expression. Once the morpholinos have been injected into the embryos, we allow them to grow to stage 40 of development. Once that occurs, we fix them in paraformaldehyde in order to perform craniofacial morphology analysis. Then we remove their heads and take frontal images. We wanted to quantify these defects so that we have quantitative data in order to examine differences between the controls. So the 4 parameters that we looked for were midface area, which is the area from the top of the eye to the top of the cement gland, and the cement gland is located right here. Facial width, which is the distance between the center of one eye to the center of the next. Midfacial height, which is the distance from the top of the forehead to the top of the cement gland. And midface angle, which is the angle from the center of one eye to the top of the mouth to the center of the other eye. For example, the Lowry lab has previously performed single gene knockdown of the 4 WH-associated genes. Here we show representative images of each of them. We found that WHSC1 knockdown caused a significantly greater facial width and midface area, while knockdown of the rest of the genes caused the opposite effect, a significant decrease. Additionally, only knockdown of TAC3 caused a significant increase in midface angle. Moving on to combinatorial knockdown, we found that knockdown of WHSC1 and WHSC2, knockdown of WHSC1 and LETM1, and knockdown of WHSC1 and TAC3 all caused a significant decrease in facial width. Moving on, we also found that knockdown of WHSC1 and LETM1 caused a significant increase in facial height. Lastly, we found that knockdown of WHSC1 and WHSC2, as well as knockdown of WHSC1 and LETM1, caused a significant decrease in midface angle. To summarize, I'd like to reiterate that WHS-associated genes are in fact expressed in migrating neurocrest cells. Individual gene knockdown has been shown to cause defects in migration as well as craniofacial defects, and combinatorial knockdown has been shown to lead to several genetic interactions, particularly those that affect craniofacial morphology parameters. We have shown that these genetic interactions are quite complex, but it is reassuring that there is in fact a relationship between these 4 genes and the WHS phenotype. Although I will be moving on from the lab, the Lowry lab will continue to study these genes in depth to ultimately determine what the genetic interaction between these genes are. They will also perform combinatorial knockdown of 2 genes, 3 genes, and all 4 genes. And this is because in humans, Wolff-Hirschhorn syndrome is highly variable, the mutations are highly variable, so we want to see how the different combinations of deletions or duplications may ultimately affect the phenotype. So this is why we have to work towards ultimately like how the loss of one gene ultimately leads to which particular phenotype, and determine how that affects to another. This is only the beginning of studying Wolff-Hirschhorn syndrome. I am confident that even though a lot more work has to be done, I think that this research will bring us closer in ultimately improving the lives of those afflicted with Wolff-Hirschhorn syndrome. Based on the results we have obtained so far, we have planned our future directions accordingly. So currently we are performing combinatorial knockdown of the 4 genes, and we will continue to do so with all the combinations. We will perform cartilage staining of in order to determine how gene knockdown affects cartilage formation. We plan to target future experiments to those that show the most robust phenotypes. We will also be using CRISPR as a complementary gene knockdown strategy in addition with morpholinos. We will also be performing half embryo combinatorial knockdowns in order to see the effects of neurocrestin motility in vivo. And we will also be performing neurocrestin motility assays to see its migration in vitro as well. So these past 10 weeks have been a whirlwind of learning, teaching, and a lot of immersion for me. And I've learned a couple things along the way. So I've never worked in a cellular biology lab before, and it was a really great opportunity for me to learn about a lot of new techniques, to both witness and perform them. And these include embryo collection, fertilization, and injections, whole mount in situ hybridizations, neural tube dissection, along with a lot of craniofacial morphology analysis as well. I've also never worked in a lab with such a large amount of people, so it really was a great way to meet new peers and learn about their experiences as well. And we also had a couple of high school students in our lab this summer, so for me, it was a really great opportunity to tell them about my experiences thus far and also explain and teach the techniques that I only recently learned. And lastly, most importantly, I really enjoyed learning about a new research topic, especially in a really collaborative and innovative setting that Boston College is. As an aspiring physician, I found it really interesting to learn about a genetic disorder, specifically one that affects so many thousands of people, and to see research being done today that may one day positively improve their prognosis is a really good thing. I'd like to thank all members of the Lowry Lab for their help this summer, specifically Rachel, Fred, Megan, and Eric for all of their help. And of course, I'd like to thank my wonderful PI, Dr. Lorianne Lowry, for all of her help this summer and all of her words of encouragement. And I'd also like to thank our primary sources of funding, the March of Dimes, American Cancer Society, NIH, NSF, along with the entire RU program. Thank you very much for listening, and do I have any questions?
Questions? Yep.
Yes.
Could you go back to one of those first slides? You had a little diagram on there. Yeah, that third slide.
This one?
Yeah, can you explain that picture a little bit?
Yeah, so basically what this is showing is the 4 genes in our DNA. So DNA is a double-stranded helix. So on the 5' to 3' strand, it shows that that's the strand that has TAC3 and WHSE1, while the 3' to 5' strand has LATAM1 and WHSE2.
Okay.
Any other questions? Yes.
So, how do you know that the frogs you're using are a good model for how this happens?
So this phenotype, so frog Xenophis laevis is used as a model for investigating gene function in general. Several studies have showed that the phenotypes that it shows is similar to what it shows in humans, and especially since we're looking for craniofacial morphology parameters, it's very obvious in their faces.
I had just a quick one.
Yeah.
With the image analysis, do you like sit there on the computer and say this point, or do you use some?
Yeah, so it's kind of like eyeing it, but again, it's mostly the same person does do the same imaging, so there isn't a lot of deviations amongst the quantitative data.
Okay, if there are no other questions, let's thank
Thank you.